Hello, my name is Minwoo Ha, and I am a Doctor of Pharmacy student and undergraduate researcher. Today, I'm presenting the work we have done on the SARS-CoV-2 replicase complex at Dr. Khaled Barakat's lab. SARS-CoV-2 is the causative agent of the COVID-19 global pandemic that has affected all of us for the past year. As a positive sense single-strand RNA virus, it makes more copies of its genetic material inside its host cell using an RNA-dependent RNA polymerase complex. Inhibiting this complex has been shown to be effective in the management of COVID-19 symptoms, as seen in the case of remdesivir, which is currently being used clinically in hospital settings as a ribonucleoside analog that competitively inhibits the catalytic sites of the assembly. We set out to further study the poorly characterized polymerase machinery to provide some foundational data and identify novel allosteric sites associated with the complex. We hope that it can serve as a basis for future rational drug discovery, namely one that targets a site other than the main catalytic site. Our lab is a computational lab. This means that we will be conducting in silico analysis using supercomputer resources provided to us by Compute Canada. We started with recently resolved cryo-EM structures of the complex and performed error correction techniques to fix any poorly resolved or known to be erroneous parts of the structure, such as incorrect protonation states at physiological pH or incorrect metal coordination. From here, we were able to generate different systems for simulation with each conventional ribonucleotide either bound to the catalytic site or already incorporated into the nascent RNA strand. We also looked at the binding mode of remdesivir in the active site as well. These simulated trajectories over 100 nanoseconds of simulation allowed us to perform data analysis, which led to various insights about the mechanism. We were able to compute and identify the hydrogen bonding networks as seen in figure 2 and electrostatic maps for each system as seen in figure 3, showing us how the nucleotides and RNA strands are oriented and stabilized for incorporation into the nascent RNA strand. As well, we were able to use correlated motion analysis and druggability analysis to identify 3 novel allosteric binding sites, which we termed the hairpin pocket, pocket 2, and pocket 3. In figure 5, you can clearly see the highly correlated motion between pocket 2 and the active site in the heat map. This is especially promising because this demonstrates that targeting this pocket can cause a dramatic effect on the catalytic sites and potentially the activity of the replicase as a whole. Our lab is the first to study in depth the structure, function, and dynamics of the SARS-CoV-2 replicase complex using the latest cryo-EM data. However, it should be noted that as an in silico structure study, it is difficult to assign clinical significance at this stage of research. However, our hope is that the preliminary data gathered here can be used for future drug design to develop new interventions for COVID-19. Thank you very much.